Good morning, everyone. So welcome to the third IAD Distinguished Speaker Series on AI and Health Science. The first two have been very successful. So yeah. So for those who do not know me, my name is Yijun Sun. I'm a professor of bioinformatics at the Department of Microbiology and Immunology. Okay, today it's my honor to introduce distinguished speaker Dr. Zhongming Zhao from UT Health, Houston. Dr. Zhao is a leading figure in the area of computational genomics, precision medicine, and bioinformatics. He is University Chair for Precision Health, the founding director of the Center for Precision Health, and the Vice President for Cancer Genomics at UT Health. So before he joined UT Health in 2016, right? So he was England endowed professor of cancer research at Vanderbilt University and the chief bioinformatics officer of the Vanderbilt England Cancer Center. So Dr. Zhao has broad interest in bioinformatics, genomics, data science, AI, population genomics, and precision medicine, and has co-authored more than 550 publications in those areas. Now, his work has been continuously supported by NIH, NSF, state, and research foundations. He has secured more than 100 grants totaling over $180 million. He has trained more than 90 students and postdocs. Among them, 29 have become a faculty, including department chair and assistant dean. Dr. Zhao has received numerous awards and is a fellow of ACMI, FAMI, and AIMBE. So today, Dr. Zhao will talk about AI, computational genomics, and precision medicine. So the title of the talk is The Road to Precision Health, Harnessing Big Genomics Data. So welcome, Dr. Zhao, to the Buffalo.
Okay, thank you, Dr. Sun, and I'm very happy to present some of my works here. This is really my honor to be the third speakers of your AI initiative in your IAD. So actually, I realized this morning I need to introduce the biomedical informatics program in my school. But I can verbally talk about it before I talk about the research here. So I came from McWilliams School of Biomedical Informatics. We actually, from size, we should be the largest. We have 3 departments in the biomedical informatics program, and we have many faculty members, and also the program covers 3 areas: clinical informatics, AI data science, and the translational bioinformatics and system medicine. So today I decided to use a little bit higher level title, but I mostly will probably focus on Alzheimer's disease, which is a very important disease, and currently one of the best funded research projects in my team. So this slide actually is quite old. I still like it because I often try to introduce my career, what I have done in the beginning. This slide still summarizes what my career, how I do my research. So for a long time, people are working on the DNA, called the genome, and the transcriptome, like microarray gene expression and RNA sequencing. And nowadays we do the single cell transcriptome, like a single cell RNA-seq, and a spatial transcriptome as well. And later you can do proteome, metabolome, microbiome, like Dr. Sun is expert, and epigenome, and exposome. So my lab actually, we cover all this kind of multi-domain, multi-omics area. And recently we also using those kind of discovery linked to the clinical data, do some those kind of clinical retrospective clinical trials to see whether some drug we predict for the specific subpopulation potentially can benefit the healthcare. So with that, I'd like to also provide some, summary of my lab. So as Dr. Sun mentioned, my lab has been productive. I choose this name, eventually now become my department name, called Bioinformatics and System Medicine Laboratory when I worked in Vanderbilt. And this lab became actually very successful, not only just because of the productivity, but also we made a lot of contribution to the medical centers through the bioinformatics, AI, or computational biology support. On the other hand, we trained many talented people. Many of them became leaders in this field. With my slides, basically I summarize 4 areas because the lab is always very big. So we developed many bioinformatic or computational approach to study, like in cancer, actionable mutation, which the mutation have a therapeutic effect. Especially when I worked in Vanderbilt Ingram cancer, we had quite a good number of projects focusing on like a cancer genome and find those mutations with a potential like a kinase inhibitor to treat the patient. And but from large scale, like cohort study, you don't have this lag to find one mutation and you have a drug to treat the patients, like Alzheimer's disease, like a mental disorder, like some cardiovascular disease. Usually it's a risk of the variants to the disease. So you need a cohort study using those kind of genome-wide association study. And then when you have many more patients, many different phenotypes, when you have big data, like the EHR data, you have the both genome data and you have phenotype data. So people develop GWAS, FinWAS. My former colleague, Josh Stanley, now is all of us CEO. He actually one of the first to propose this GWAS, FinWAS kind of idea. And then you can link this to EMR, become a very synergistic kind of approach to mine large-scale phenotypes through the genotype data. So it's more like one is like 1 to N and N to 1. And then you have N to N kind of a dimension to study the genetic variants and their roles in disease. And during the last 7, 8 years, we're also very active on single-cell omics. And later I will show you some research project. And we're also very interested in the pharmacogenomics network through the drug target and the biobank mining and do the drug repurposing. And nowadays, I have a good number of active grants, except all the grants we have deep learning in the keywords and to use deep learning approach to study genomic features and the drug response. So this is also a high-level summary. My lab actually developed many methods and tools as well as database for single-cell omics. I list a few here. And also deep learning, we develop those AI-driven like Genomic, and also we study virus integration studies, deep learning. And we also have I also will meet the leader from dental school. For dental, I have several grants working on like a craniofacial development. One is called deep face to using deep learning approach to find the genetic variants potentially with a low in those kind of craniofacial development, linking to the oral like cleft lip and palate. Because of time, I just very brief mention here. If you're interested, I have a lab page you can visit. So from here, I probably go through several projects. That's as a bioinformatics or computational person. Basically, you can ask me any question, and I can stop anywhere because we are often, data-driven, algorithm-driven. It's not like a traditional one disease. You spend like 20 years, 30 years working on it. So I just use several projects here to demonstrate how the bioinformatics lab potentially we can help like laboratory scientists, physicians, or some other people to better mine the data and to find the potential, the generate good hypothesis. So this is a little bit old. We use the those large scale, those kind of brain sample, over 500 samples, and it's microarray gene expression across the human brain called the atlas of the human brain called the brain span. And the data is often, as I mentioned, summarizing in the right part, is often heterogeneous and high missingness. Those kind of sorry, I cannot show here. So this kind of white region usually is data missing or low quality, and the blue area cells are good quality. So we use got a development method called a Bayesian tensor factorization model. Basically, it's a multi-dimension matrix, and through this kind of the tensor fractalization model, you can identify those features and then predict the gene expression in the specific brain region of the specific human samples. And this human sample covering the prenatal to postnatal, including like adults. So it's through this work, basically, we can better inform the gene expression in the brain region across the brain ages. Okay, so then here I like to, I'm very interested in the Alzheimer's disease, so I like to use Alzheimer's disease to demonstrate some work we can use AI and related like genomics integrated by informatics to study the AI as well as some other disease. A lot of approach actually can be similar to apply to other complex disease like cancer, like a cardiovascular disease. So AI is a very complex disease. It's usually occurs in older stage, like 60 years old or later. And it's usually called, the name is dementia. And the dementia, a majority of them, like 60 or 80% are called Alzheimer's disease. And the Alzheimer's disease usually occurs after the 65 years old. This is called the late onset Alzheimer's disease, the majority of them. About 10% are called early onset, which means those patients, they were diagnosed before, earlier than 65 years old. A lot of argument, whether I'm not an expert, by the way, these two diseases, early onset and late onset, are two kind of disease, not necessarily the same, the same biomarker, same mechanism. So I'm also very interested in whether using genomics can study early onset, whether it can be unique or whether it can be better linked to the late onset. Today, I just decided to take some slides here to talk about this. So late onset, early onset, again, is very highly heterogeneous. The heritability is actually pretty high. And also, on the other hand, because of the environment factor, make the data can be noisy, not easy to find the genetic markers. So with that, recently we looked at some grants, and fortunately, almost everyone was funded. So one of them is the U1 funded about 2 years ago, called AIM-AI, actionable integrated and multi-scale genetic marker for Alzheimer's disease through deep learning. So in this algorithm, we try to first develop an AI algorithm to study, using single cell and bulk tissue RNA sequencing, mainly focus on the epigenomic data to develop AI algorithm to inform those kind of AI susceptibility risk, whether have a better risk to the AD. And then M2, we use the neuroimaging. Sorry, it looks like because of use different computer. So use neuroimaging and the AI algorithm to identify those patients with specific features linked to the AD and the progression. And then we try to link them together, better inform the imaging, the feature can better identify the markers, and the markers can better inform the neuroimaging features. And then we use Rush University Medical Center data. They have longitudinal multi-omics and imaging data, and can validate our finding. So that's kind of the idea. So with that, we have developed several sub-projects within this U1, like neuroimaging genetics, our main focus. We also study polygenic risk score from genotyping, and to find whether they can better inform the genetic variants, a set of genetic variants with better power to predict the phenotype outcome. And we also study those kind of, the AD related to psychiatry or immune disorder with shared genetic architecture. And also we use single cell omics data to study AD and the subtype, like early onset versus late onset. And eventually we want to study multi-modality. Now we use modality versus domain across the multi-type of data, and use transfer learning to learn the feature and inform another type of data. And even another type of data has a limited power. We can better inform the features and help them to mine in another domain of the data. So with that, I just gave one pilot study in this U1 to study, overall how the neuroimaging genetic data can be linked to the genotyping data, and how they can help each other to have a better power to predict the potential variants involving the Alzheimer's disease. So this paper is published last year called Distinguishing Accelerated Cognitive Decline from Normal Aging Process, which is unique from the many other studies. They usually starting with the cognitive decline, the disease sample. Here we use a normal aging sample. And then what we learned the feature, we inform the Alzheimer's disease patient data, and then try to identify the genetic information linking to those kind of IAD phenotype. So this is a schematic overview. So the data we used is ADNI, Alzheimer's Disease Initiative. It's a longitudinal study, have both neuroimaging and genotyping. The sample size, unfortunately, is still relatively small, about 1,000. So we basically developed deep learning to study those Alzheimer's disease imaging, the normal brain cognitive decline imaging data, and then compare to those Alzheimer's disease patient imaging data. And from them, we learn the features. And then from those features, because the ADNI data has a genotyping, so we find the related genotyping information, find which kind of markers can be related. And next slide actually give you more detail. But regarding the AI algorithm, this is show you the algorithm. So we have a longitudinal neuroimaging for ADNI sample. So we develop those kind of, the Siamese network, which I have slides to you, the algorithm. And then based on Siamese network, and we find the RaceNet 101 specific algorithm, we find can detect those kind of the difference from, those kind of the longitudinal imaging. And then we find the features in those brain regions. And then we come back to the genetic data, and this is GWAS Manhattan plot, and we find whether some mutation have a better risk to the phenotype based on those neuroimaging, and then we try to validate through larger scale genotype data with like over 1 million samples. And again, this first one, ADNI only have like 100 samples, 1,000 samples. So with that, the Siamese network, this is a little bit old. I shouldn't make another one. Actually, I have. This is basically you have a it's a good algorithm because we often see unbalanced heterogeneous data, and people usually use case controls like tumor versus matched normal. So a lot of time for like neuroimaging, this doesn't have enough power. So the Siamese network is also called the triple neural network. Is very efficient. So you basically use an anchor, which is can be linked to the positive data set, the sample, and then you join them, and you compare the negative. Because this linking sample, they improve the power when you compare to the negative, which is match the control. So this algorithm basically, like we also developed this for single cell, demonstrate very efficient. And this is a more professional way to show in the triple neural network as a kind of the Siamese. So basically, like imaging, you have a car, another car, but you are not very sure the related features. But when you compare to another transportation tool, like an airplane, they have power. And because of anchor linking to the positive, they enrich the signal. And when you compare to control, you improve the power. So that's kind of idea to do those kind of a triplet network. So then specific back to the ADNI, again, ADNI is the based, according to the data collection, this is the best data set for Alzheimer's disease. It's longitudinal, spend, I don't know, at least more than $100 million. It's a good partnership from industry and the academic field like NIH. And the data still now goes to is continuing growing. They collect sample and do imaging and multi-omics, including recently we try to acquire the methylation. So the data again, as about 1,000 samples, and after the data cleaning, we find a control group, and the control group longitudinal with age and imaging. So we find those training data set and validation using those kind of typically separation like 2:1 or 3:1 kind of ratio. And then we use the ResNet, which is a common algorithm, and to evaluate which kind of model has a better performance to detect the feature in the neuroimaging. Again, the backbone use the triple neural network, and then we find the features and using normal imaging data, normal sample, we find the features. Then we come to the Alzheimer's disease subject, find the subphenotype, and then we come to the bottom is the genotyping data linking. It's all linked to the part of this imaging data set. And then it's about 1,000 and the European ancestry data set. Then we look at those cognitive decline, and from our study algorithm, we can predict accelerated cognitive decline because we use normal imaging data. So when you compare normal imaging, when with the AD data with normal imaging, we call it accelerated cognitive decline. This actually improves our power, and then we find a genetic marker. We study the markers with Manhattan plot and the polygenic risk score, as I mentioned a little bit early on, and functional interpretation, and also further genetic study like a co-localization with other disease. So because of time, I don't have a slide to show all the detail. But the one thing I can say is using our algorithm versus the traditional cognitive decline slope, we use our algorithm called accelerated cognitive decline, which uses triple neural network for aging, normal aging, which has better power. So we were able to first validate this well-known the ApoE locus on chromosome 19, but also we found some loci which is promising. One of the locus is above the genome-wide the significance on the chromosome 11, and this SNP is in the NAIR1 gene. This gene is very important regarding synaptogenesis, neural outgrowth axon guidance. Actually, when we shared this study with ADSP in NIA, the former program manager was very excited, asked us, say, there's another cohort found this SNP gene looks like very interesting in Alzheimer's disease, trying to validate through their work data. So again, if we use the normal, like a case versus control, find a cognitive decline slope, this actually couldn't find those kind of marker gene. But use our accelerated cognitive decline based on our algorithm, we were able to identify this SNP and the gene, and then now we do the follow-up downstream like validation, and we use those kind of co-localization for the additional gene regulation. We found that this locus actually very interesting. So because of time, I'm not going to give detail, but as part of this project, as a conclusion, so our deep learning method based on the normal brain aging, called those kind of normal cognitive decline. Then when we compare to IAD data cognitive decline, we were able to improve power to find those imaging features linking to the cognitive decline in AD. And then through this linked genetic data, we find some locus, especially locus in NAIR1 gene, potentially can be better explained in the genetics of AD. And this approach, potentially we can apply to other disease, especially for AD, if we can find a larger cohort with brain imaging, both cognitive normal aging sample and match the AD sample, we can improve to expand to the other data set. And here, because this is an AI seminar, so I like to put another study related to AD. We recently developed called a brain gene bot, because everyone now is doing GPT-based, to help your research or daily work. By the way, maybe I just do a very quick search. How in the last week kind of range you used any of those kind of GPT online tools like OpenAI, like Gemini, like my favorite one is Perplexity AI. Raise your hand. Okay, almost. How many of you actually use this in your writing, regardless grant manuscript writing in the past month, within 1 month? Okay, only a few. How many you think you should use OpenAI, those kind of tools, help you write grant manuscript? Okay, very few. Actually, I involved several discussions recently whether we should, the third question, whether we should do it or not. I think it's arguable. And NIH actually released a guideline using very interesting. They will administratively withdraw the grant application if they find your proposal substantially use AI. How you define this? My associate for research said, how can I interpret this? So we said, use AI to get your answer. So, but on the other hand, I think I had 3 steps in my lab. Basically, about 2 years ago, basically GPT was released in late October 2022. Within half a year also, I said we have to be extremely careful because I was originally trained in PhD program. I'm more traditional scholar, this kind of training. So I said, you have to be very careful. I don't want to have any paper, people say you have some error or misinterpretation. And then about 1 year ago, I said we have to be cautious, but why not use GPT? When you write some draft text, why not go there to enhance it? Especially some international students, trainees, English may not be so as a native English speaker, including me. So I found it very helpful. And I also bought like a Copilot. It's a small amount of monthly fee. Now I write email. I also see half a year ago and now the tool become much better. Now even 2 or 3 sentences, the Copilot can help me to write the email response. But before they said, okay, you need to write more. They do it. So now I think I more tend to do the third one. We should use AI help our write, but you cannot only rely on it. You have to job responsibility. Your effort change to when you write something. Obviously, you already have some idea. You have to draft the text. You have to very carefully annotate your writing. That's very important. On the other hand, AI now is not just writing. If we are good at it, now can generate a lot of images, videos, but also they can, like Copilot, I got some training 2 months ago, 3 months ago. Basically, they can, you have some idea, write some text. They can propose the draft the slides for you. It's actually pretty cool. I shared the slides during my training to my lab members. They said, wow, that's amazing. So one of my projects, they actually use those slides as we are develop a database for brain, use another image because they found it authentic. That's Copilot. They said all the image, they have a source, not artificial. So we actually use that image for our database because we have a license. You can use just like Adobe. Okay, but with that, we actually very quickly follow this, even though it's still relatively small effort. Use GPT to develop some agent to help us not only lab but also community to better interpret or better mine the data. So one of the project we call a branching bot now. Because it's basically, we are very interested in genetic signal. And there are a lot of data, a lot of GY signal, and very noisy. And many people just manually look at it. And they require both genetics, but also like a phenotype expert, like a physician, to help each other. So basically, this idea is why not we create a better customized engine. People don't use programming. They can write a plain text and go there, find the information, and better interpret. So this is our one of the efforts. So we call it branching bot integrate rank aggregation with GPT-informed polygenic score analysis. And because of our grant and many grants now in Alzheimer's disease, we're just demonstrating Alzheimer's disease. So it was initiated by Annie Nas. She's a PhD in computer science, and now gone to a postdoc also in bioengineering. So I intentionally put this figure here. It's not perfect with some typo or something here. So the reason I put this is I have to remind me and my team worker and also users. It's a tool potentially give you a big help, but never trust everything from this kind of tool. That's very important. So as a bioinformatics faculty and a leader for over like 15 or 20 years, so I often told many people one thing. People say, wow, you did a great job. I like your work, blah, blah, blah. I often tell them, yes, thank you, but always be careful. We are bioinformatics. We help you, but we are not like give you all the solution. You have to make sure something may be wrong. Either our method potentially can have false discovery, or potentially I give you something, you actually ask it something else. For example, you have more specific focus on a subtype of the tumor, but we actually give you all the whole kind of phenotype of tumor. The interpretation will be different. So and also power analysis, sample size is always one issue. So with nothing here, like a rank aggregation, it's a long one. But if you don't look at it carefully, you must like it. This is a very good figure, right? So with not a little bit background, genome-wide association study. So old days use a chip, the microarray, now the sequencing. You have a large number of cases. Nowadays can be usually several million, and control match the phenotype. Like Alzheimer's disease, you have the case and the control, and then you need a the genome-wide p-value. The nominal is p-value is like 3, but you can genome-wide you need a 10 minus 8, and when you log p is basically 8. So this is a very standard the genotype association study. And then you have Manhattan plot. Each bar here is one chromosome. Each dot is one SNP. And this log minus log P, you need to reach like 8, reach the genome-wide significance. So with that, long time, people spend hundreds of millions of dollars generate many genome-wide study of the data set. And often they report only one or few, and many of them, the SNP actually in the non-coding region. That's the open challenge, how we find the genetic marker involved in disease. Then with part of them, people propose a polygenetic score, which is statistical modeling. They put they use a seed marker, just an index marker with a significance of the signal in the genotyping and genome-wide association study, then expand by linkage, and then they put this together as not one locus, several or many locus across the genome. Or together, they have a set of SNP called that's why I call it polygenic. They overall contribute to the risk to the disease, much better power. Then the PGS has been there over I guess 15 years, and then no surprising this database called a polygenic score catalog. Unfortunately, the good news is they have 600 trees, 5,000 polygenic score data set, and many publications. And this is a kind of backbone how you do this kind of polygenic score. Because of time, probably I don't talk with detail. But interestingly, or challenging thing is when you search those polygenic score, for example, Alzheimer's disease, it came up like over 50 data sets. And when you look at those data sets, supposedly when you look at them, you should have a conclusion. Oh, those kind of SNP as a whole polygenic score can inform their risk to disease. Then your work is done, right? But unfortunately, when you look at those over 50 data sets, some only like less than 100, some like over 1 million. So take home message. I don't know how to interpret this. This is very, very heterogeneous and very tough, challenging to interpret. So what we do basically, we try to download those data and develop those, those kind of firstly download data, streamline data from the online and develop engine, and then we do the data analysis too for the downstream. Not only look at the SNP as a set, like a polygenic score, but also to interpret like through string, like gene set enrichment, enricher, and ClinVar for the clinical assessment. And then real-world application, dynamic, API, real-time, and customized. You don't need to write a program, just write a plain text. That's our idea. And more specifically, there are many SNP, many data sets. You cannot just, put them as a summary and all make a very simple weight-based, if you see this SNP in over 50 data sets, you think this should be true. Not necessary, right? So then we use an algorithm as part of the backbone within the engine called the rank aggregation. This algorithm actually has been very popular for like 20 years, but not many people actually use it daily. So basically, you have a sort of, a list of gene or SNP, like a gene expression across the genome, and each gene you have some impact in the disease. So you basically, based on the p-values, typically you rank them. But if you only have one data set, it's easy to rank by p-value, right? What about if you have 50 data set or 20 data set or 5? So they have many, within rank aggregation, try to integrate multiple data sets. They came up with like 4 categories. This is the review article we published recently. The easiest one, like a Markov chain, probability based. And we also can use like a Bayesian model. So they have about 20 algorithms there. So we choose about several of them. We just evaluate and implement in our algorithm. And plus the data set, as I mentioned, some data set only have 100 SNP. The other is like over 1 million in the PGS catalog. How you make those unbalanced data, put them together. So we need some machine learning to take care of those unbalanced data set. So my postdoc actually came up a very interesting but powerful algorithm called the transactive learning. This one I just can put here. So one is we use rank aggregation to aggregate all the evidence among like a large number, like 50 PGS data for AD. The second one, those the data set are very unbalanced. We use transactive learning. The traditional one is you have those, the positive one, negative one, and you just compare them to them. The transactive learning is very interesting. It's it can, doing those kind of analysis, can, if we have 50 data set, right? For some data set, it can border those kind of, different label like case control, border some control to the case or vice versa, do the swapping and do the analysis. This is essentially just like a triple neural network. You increase the sample size and do more multiple comparison. He actually convinced me this actually worked very well. So with not the algorithm, not only just the GPT engine, we implement those kind of rank aggregation. We develop an algorithm to deal with the highly heterogeneous, unbalanced data set. So that's kind of algorithm. So I probably skip this. So this eventually we have this branch gene board. So the first part you do the user interface. You just use plain text. I want to find the genetic variants associated with Alzheimer's disease. Or you can say, okay, I want to ask those early onset or very old stage or very severe, those kind of later stage of the progression of the Alzheimer's disease. If we have data from the data set, it can inform it. But the underlying, the rank aggregation to aggregate the data from PGS catalog, and then downstream, once you have those kind of ranked list of variants, we implement the API like a protein in network, like a gene set enrichment, like a GTEx is a tissue gene expression signal, and the ClinVar is for the clinical assessment. We also implement those, the deep learning now algorithm to mine the literature from the PubMed and all together. So basically, it's like a one shop for everything, starting with a plain text to ask the question, like you go to the OpenAI. Came up with a list of rank aggregated variants and the downstream interpretation. So we are still, developing this one. Hopefully, we can expand it to other like GYs or other, gene expression data set. But I think this framework potentially can be useful for the physician or laboratory scientist. So, okay. So from here, because the AI institute here is also try to for drug discovery for biomarker. So I use the third project basically talk about how we use this kind of informatics approach to for the purpose of collaboration or exchange some idea learn from you. We developed this kind of informatic approach to potentially identify the marker gene and potentially for drug target. So I just talk about some project in this area. This back to the early days, my lab systematically used those cancer genome data from TCGA, the Cancer Genome Atlas, and then I was very lucky in both TCGA and this International Cancer Genome Consortium. And ICGC published a series paper in Nature and the Sister Journal in 2021, about 28 papers. I was very lucky to author many of them. And again, this is a very big consortium. It involves the milestone project is 25,000 whole genomes, tumor genome, and 50 cancer type, and the many SNPs. And my lab using bioinformatics, genomics approach to basically mine a lot of features in cancer genome. And each of them, we are very lucky to publish some papers, like a positive selection, mutation hotspot, like ligand binding, like a phosphorylation pocket region, and a gene fusion. And nowadays, obviously, use deep learning. I didn't put one here. Is we use deep learning to predict the drug response in large scale, cancer cell line drug response data set. But I like to talk about very briefly mention is we use deep learning to potentially identify those kind of B cell epitope, which potentially involving immune response with this called the immune app. The idea is actually very simple. In the database now, we have many those kind of B cell and also T cell. We are doing that those kind of peptide, and they have those kind of binding affinity data set. However, as you know, those kind of HLA region is very variable, very hard to know a specific pattern. And another important feature is those kind of small peptide, usually like 21, 22 amino acids long. It's very because of short distance, it's very hard to do modeling because you need usually sequence count is longer the better. So with that, the deep learning potentially can help identify those kind of hidden features and then use, the like weight matrix or some other potentially can better inform the feature mining discovery. And we eventually apply this to the HI epitope and find those features and better predict. Because of time, I just mentioned very briefly. Actually, the first author knows more than me in this specific topic. From here, I also like to talk about a little bit single-cell RNA sequencing. We since 2018, 19, we are very interested in single-cell because we are always doing genomic medicine, genomics. So my lab actually developed several algorithms to deal with the data missingness, like MTNN, and the batch effect called the insight. And we also try to use single-cell to find the driver and the mitochondrial variants as well. Driver basically is a transcriptional program of the key regulator in the cell-specific manner. And we also develop several databases like WebCSA to identify a set of gene or variants, whether have enriched in specific cell type. This is very easy to use. And we also develop a single-cell GYs. And recently we have a polarization using single data set. Any single-cell data set, we can predict cell polarization. And I have some brain disease kind of single-cell database, and this is for Richard transposon element published last year in Genomic Research. So here this morning I actually changed some slides back. So I like to use not AI, but to talk about some single-cell modiome potentially can help us to better interpret the disease like Alzheimer's disease. I saw the audience may be more interested in the discovery, not just AI algorithm. So I decided to move some slides here. So single-cell modiome is a very important technique. It became the Nature method in 2020. In the past, like 7-8 years, Nature method has basically every year they nominate the method of in the field, and most of them are related to sequencing. So this is very cool. Like last year is the long read sequencing. So single-cell modiomes is a very good technique because through one experiments, it can generate both single-cell RNA sequencing, which is gene expression, but also the epigenomic called single-cell ATAC-seq, which is chromatin remodeling. So one experiment, the same sample, generate both type of data. This essentially remove the batch effect through the technical part. So we actually applied this modiome in several projects, including craniofacial development. Here we apply to the Alzheimer's disease. Because we don't have a large budget, also we are, earlier, we are not expert in AD, so we decided to choose a unique phenotype called early onset Alzheimer's disease, because there's no such kind of study in early onset, like single cell, especially modiome. So obviously it's not easy to identify the samples because this post-mortem brain tissue. So this is a very small sample but it covers, each individual covers 3 brain regions: prefrontal cortex, entorhinal cortex, and hippocampus. And we have matched the control by age, sex, and the other phenotype. And then we apply this single-cell modiome, both expression and epigenome like ATAC-seq. And overall, we generated about 76,000 nuclei. And this is the overall the major cell type distribution. And at the bottom here showing the cell gene expression, single nuclear RNA sequencing, and ATAC-seq. Overall, the quality is very clean. And then we do those kind of analysis. Then what we do first, we do those kind of in each brain region, which kind of differential gene expression, downstream, down and up regulation in each brain region when you compare to the match the control. But obviously, we also want to do the regulation because you have a modiome, the epigenomic change. So the idea is like, okay, if you have a genetic variant and the ATAC signal, and you have gene expression in another locus, how you can connect them. That's called a candidate, those kind of candidate cis regulatory element. So we did those 3 brain region, the distribution, and here is showing those kind of distribution of the cis regulatory element in the 3 brain region when we compare to the different those kind of encoded category, we see, for example, the distal enhancer like signal is a very large proportion. And also we see those kind of a promoter like a signature, very large portion. Provide an overview of those kind of regulatory element in early onset Alzheimer's disease, how likely involved in in those three brain regions. Obviously, we can go deeper level to see those kind of regulatory like a transcription factor, the regulatory gene, and because we have three brain regions, we can also see brain-specific regulator and a consensus regulator. And for those three brain regions, all we see identify those transcription factor. We found 7, and those some of them very interesting to us, like IFX4 and IKZF1. So this is showing the specific cell type in entorhinal cortex and how those kind of gene expression regulation involved. And this is the gene regulator network, highlight FX4. It involves in many other gene and regulator. And the different color means the transcription factor and the gene, and the red is up and the blue is down regulation. So this again is we also find a motif of those kind of transcription regulator because it's a higher level. Many figures I just show some overall figures, and this is essentially we need a validation. And unfortunately, we did some experimental validation to show the evidence. And then because I have a very strong background in genetics, so we go through the genetics like to see those kind of GYs of the Alzheimer's disease for those kind of regulator like the differential express gene and also the CRE, the regulatory element, whether they have enriched genetic association signal. And eventually we find those related disease, whether they share the specific regulatory information from our data set. Again, because of time, I just very briefly mention it. And then we do cell-cell communication. So as summary, I just very briefly mention. So this is the first single nucleus multi-omic project in the sporadic early onset AD, covering 3 brain regions. And we find some interesting candidate regulatory elements, and also some transcription factors. 7 transcription factors are commonly shared in the 3 brain regions, and they are mostly interesting in the glia cell. And we also try to compare the early onset with the late onset, and also we find those SNP related to this data set that can be related to other, the phenotypic, the brain disease. So because of time, I just go. I think I should skip some, those kind of, the immunotherapy using single cell. We find those target, and we also do the recent method for cell using single cell foundation model to identify the polarization. I just skip this. Okay. So I like to take maybe 3 minutes to talk about something overall from my very unique experience in bioinformatics field, not AI, to share with the bioinformatics field, how we go, where we come, and how we go. So I always consider I'm a traditional PhD, trained PhD. I'm not like many of my colleagues. They are computer science. They basically say, let's do it. Sometimes even they don't understand what it is, like Alzheimer's disease. I'm always trying to understand what is the problem we need to solve, how we can help understand the disease, how we can help the healthcare. So this is actually, the reason is because when you look at this, that's my PhD training. Obviously, I'm not like so old, but my PhD mentor and his peer, they are talking about those kind of, the traditional, the algorithm, population genetics, molecular evolution. You think about those kind of model like, PAM, which is those kind of mutation weight matrix. They use protein sequence back to 1960s, 70s. Or early 80s. They only have a very small number for protein sequence. They already developed an algorithm. And later, when the genome sequence became feasible, especially human genome data became available, you find that those algorithms all become feasible, and they already developed elegantly there. So one of them is a unit blast, right? So blast algorithm, the local search is all based on those kind of early days, the models like PAM, especially protein blast P blast, okay? So my PhD mentor is mostly famously drive the field back in this field. And I consider 2001 is a milestone. Why 2001? Because when we do AI or omics, we need the data. So 2001, February, I believe the 11th, published 2 papers, one in Nature, another in Science, the human genome draft sequence. So from there, the genome sequencing becomes more feasible. The mass of the data becomes meaningful. So before this mainly methodology, it's a build up the field. And from 2001 to 2022, which I consider GPT and the single cell became mature that time, and the GPT released in October, late October, I consider that this is second phase of the bioinformatics. I consider this actually helped me to grow my career mainly. I benefit from this period. So you see a lot of human genome sequencing, next generation sequencing, third generation sequencing. Nowadays, even though few people mention third generation sequencing, and the multi-omics integration, like my first slides, and the cohort network, like a GWISE network, and a knowledge base, a lot of data built up. And this basically is a resource building. And with this kind of resource building, not only data, but also the knowledge, eventually help us go to the third level, which we need a large language model, we need the GPT, we need an AI algorithm to deal with those kind of mass amount of data. On the other hand, it's not just doing this. You need to be careful whether we should use the FAIR principle, make those kind of the data mining more reliable, accessible, interpretable. And finally, when we're working in the biomedical field, I consider our final goal is make it translational. It's not just make for fun, for paper. It should be useful for the patient care and useful for the drug discovery. So this is again very high level, very personally naive way to think about the 3 stages of bioinformatics, but I like to share with you. So with that, I like to thank many people in my lab, especially those highlighting the board here. They made a major contribution to the talk I mentioned here today, and also many collaborators. And for Alzheimer's disease, I have a key collaborator, Xiaojin Jiang. And we also were funded by grants. And this is before I left the Vanderbilt, everyone except one who went to industry became faculty, and during COVID, and this after the COVID. With that, I stop here. Thank you for your attention.
Any question?
And while I'm waiting for the question, I also like to share with you this one. Thank you. This is some photos I prepared from my lab.
Thanks for the talk. It's very great. So yeah, I noticed you mentioned the translational. It's a big thing for the next generation of the bioinformatics. So I wonder, what do you say how to bring this gap? Because I found that in my practice, it's easy to design new models, right? But sometimes it's difficult to bring these models link with the interpretation, with the translational. So what do you see in this challenge? How to deal with this?
Right. Actually, in general, not only AI for medicine, not only AI for biology, this is a common question. The AI is algorithm, especially with now easy to use, means it's more likely to have a false discovery. Very simple. 2 years ago, everyone is talking about, okay, one interesting thing is because my background origin in China, right? So people ask, what is the Fudan University? Many of you heard about this online chat. Say, oh, Fudan is a good university. Then the person asks, why you don't think Fudan is #1 university in China? Then ChatGPT will say, oh, you're right, Fudan is #1 university in China. So this is misleading because you guided them to answer the question. So by saying that, to answer your question, yes, that's why my lab, I have 3 stages. My stage 1 is exactly like this. You have to be extremely careful. Don't think, you could answer beautiful, elegant writing, those kind of summary. It's what you have. You have to use your judgment. You have to do additional search, use your knowledge. On the other hand, indeed, AI now, I especially think about, 2 ways. One is later part, enhancement, but also regarding our real research. Another one is early days, when you have some idea, but you don't know how to formulate your research project, formulate your great idea, like form a bigger program, formulate like a national network. You came up with some idea, write some text. The AI, like GPT or others, can very quickly give you a structure-based summary. This really help you. But again, don't stop it there. Like when I ask my colleague or my program manager, say, I want to do this, like I'm running the vice president office for genomic medicine. I ask my program manager, help me get something like in topic. He sent me one day. I look at, wow. Then I look at the text, say, wait a minute, did you just do that? Said, yes. I said, you need more homework because this can easily see this is from AI only. You need your own interpretation. So you have to be very careful. Otherwise, it's always biased. It's always potentially misleading. But the bias probably more likely the current status. Okay, any other question? Yeah, thank you, Dr.
When you're working with the imaging data and discerning genotype-phenotype correlations, one of the interesting factors is that brain shape is, of course, dictated by the genes that encode the development of that organ. So when you're looking at a base set point, which is different between individuals, my question is do you control for that before you then move on to do your analysis for genes that provoke Alzheimer's phenotypes?
Okay.
Do you have a way of looking at what your true baseline is, or does it all get sorted out in the total number of scans?
Yeah, that's a very good question. And obviously this question, that's why you ask, it's a more, $100 million, because I heard ADNI is more than $100 million project. So like brain, space, like brain size, these are all very important. So collecting such kind of data, I think now still limited. For psychiatry, for example, people find the brain volume is actually related to psychiatry disorder outcome. In our model, actually, I don't think we have such kind of information. We are mainly, consider two kind of data. One is kind of like fMRI, the traditional imaging data. Second is related clinical phenotype information, not probably include a very specific phenotype, but some of them, they should have information. We're mostly including aging, age, sex, drug treatment, ethnic background, and cognitive decline score, and also some related, the drug treatment patient history. So we don't have the other subphenotype. Yeah, it's probably not easy to collect large scale this kind of data. Yeah, from AI perspective, if you have even partially, you can always put in. Then it's the potential, false discovery also. You need enough sample, high quality sample as well. Yeah. Actually, I'm very interested in the normal brain aging. Because many people use that, you think about this, normal brain aging is more likely high confidence data, assuming the phenotype is, more manageable, right? But for disease like a cognitive decline, it's very hard to measure. Phenotyping is very, very challenging. So when you put in a model, potentially it's biased, or you don't have enough power. Okay, yeah, please.
Hi, I have a question from online. They said, coming to the brain gene bot for the RAG, did you use a hybrid approach?
Hybrid approach, what does that mean? I guess maybe that person is not available. Yeah, basically this is review comments, not hybrid approach, but the review indeed asked us to do more RAG. Actually, I'm not a very good, good expert on RAG. But I think RAG, based on what I learned, now it's important 2 years ago, but now it's not so important because the model now is very well, GPT model is very well developed, not like 2 years or 3 years ago. So we actually addressed a similar question, not hybrid, but how we can better evaluate by using RAG. And I think our response to review is satisfactory. Overall, our response is RAG was important, but not used so important like before, because the again, the GPT model now much better than before. So more important now, actually, I'm more interested in is the inference AI because the foundation model now after 2 or 3 years, much better than before. How can we use inference? Not only like we are doing downstream analysis, not inference, but potentially you can use, inference AI. We can interpret here, better interpret.
Thank you.
Thank you. Yeah. Any other question? Any questions? Good. And again, don't trust me. I only have a limited knowledge, and running a big lab. That's also I ask my lab member, be careful using AI because this is not only I'm the unique. For many biomedical research lab, the unfortunate PI will not look at it like when I was a young PI. I look at every data set, look at every writing, and check the record. Nowadays, it's more like a systematic way. If you have a senior PI, the junior PI or senior lab member, they take care of the project, and the young people doing the work. So this kind of system using AI is sometimes very risky. So a lot of time I worry about the quality and the fidelity of the result. Okay.
I have a question. So, for bioinformatic analysis, right? So when we try to publish a study analysis in a high-profile journal, one question that review always ask is how to validate your results. So basically, it's kind of related. Another question is so bioinformatics often we do the basic science research, right?
Right.
And also we want to translate our result to the clinical setting, basically translation research, right? So there's a significant gap between the basic science research and translational research, right?
Yeah.
So how to close the gap?
Yeah. So that's again very good and important question. So to me, I think we have two types of the validation. First one still traditional, like the early onset that we mentioned. So the review ask experimental validation. Review ask clinical interpretation. Ask specifically physicians can help us interpret the phenotype of the data underlying the data. So that's more traditional. If you need to publish top journal, this is almost standard. If you don't, if you do large scale data-driven informatics work, if they don't ask you in the top journal, this will be very surprising. I usually say at least one review will ask. The second one is something we can or cannot do it, and we often sometimes can convince reviewer and editor, sometimes not. Is we can find a good excuse. For example, if you build up a foundation model, you say I found like a Google Genome just released like 1 month ago, or AlphaMist Sense like 2 years ago, right? So one good reason is I only have so many data sets, and it's everything I put here. My algorithm already there. So there's no way to find another scalable data set to do the validation because that's all the data we have. So you have good data. Then they ask you is basically model fine tuning, blah, blah, blah. Obviously, you can still do experimental validation for some top, those kind of top candidates for whatever gene mutation. But overall, you don't have validations for your specific algorithm. So this is, this is common nowadays. You don't have a resource to do it. This potentially risky. Anyone knows single cell GPT, SCGPT? Okay, great. So about 1.5 years ago, probably 2 years ago now, when it came out, very, very excited, right? Are you still doing SCGPT? Still using it? Probably not. And I heard many negative feedback because it's the first one. There's no data. Again, they use HCA, Human Cell Atlas. That's the only large scale across human body, all the organs. You can do that for training. But now, that time, you don't have a comparable data set. You can do the validation. But now you have more data. People talk about 1 billion cells. On the other hand, the algorithm, first one is good. But now you may have better algorithm. That's, again, the answer is only partially. This field is very dynamic. Yeah.
Okay, thanks. Any other questions? Oh.
This question is not
as much about your specific research, but sort of the big data in genomics in general. One of the things that we hear a lot about outside of genomics with AI is, the quality of the data that like something like this foundational model of ChatGPT is trained on, and biases that might exist. When we have these huge analyses that are trained on this large public data like TCGA for the Cancer Genome Atlas, I don't hear a discussion about the quality of the data that exists on there being considered. Generating like a gene expression matrix that is being fed as an input into like some of these large analyses with AI. Obviously, there's so much variability in how that data was generated, whether, especially since we look back, a lot of this data has been generated since 2001. So the process of getting that has changed a lot. Like if we look at variant calling for these large, genome-wide association studies, sometimes in the context of cancer, there's a matched normal sample, which is, a better option for, looking at somatic variants, and sometimes there isn't. Like, how is that? Is there discussion about looking at the quality of the data that is, these big analyses are using?
Right. So again, this is a very important question. I actually didn't mention why we do the brain gene board. So underlying, we are doing meta-analysis. However, this is only partially answer your question, short answer question. When you deal with that, regardless you do foundation model, you do AI algorithm, or traditional, you have a one disease, multiple study, the so-called meta-analysis. So I just use meta-analysis as a one point. When you do meta-analysis, even one gene, one SNP, or when you across only one disease across the data set, this sometimes a lab can do it, but a huge effort. But a lot of time you need a cohort. They carefully curate, annotate to from the data generation, heterogeneity, phenotype, and the data missingness. Something like Alzheimer's disease, you use say 10 data set. There's some sample in this data set, but also in another. That's huge amount of effort to how you find out, number one. How you remove it, even more challenging, right? Find out, okay, UK Biobank, for example, a lot of AD study use the UK Biobank data. But then when you remove it, how can you do the analysis again? It's a real project, right? So if one lab you do this, it's also mission impossible. You need many people. So those kind of meta-analysis paper usually highly cited. Usually you can publish in very good journal, but it take a lot of effort. So that's why we came up with gene board. It's a those kind of meta-analysis. You try to use a rank aggregation, you use those kind of transductive learning to learn the feature, do the meta. But the answer to your question is, if you really want to do a very perfect foundational model, you need all this kind of work, do the curation of the data, deal with the data missingness, batch effect, platform, across platform, phenotypic change. It's a lot of things. It's almost impossible. So people are just very close to it. On the other hand, from algorithm, we still have many things can do. So nowadays, a very common one called transfer learning. So this may not be accurate, but again, when we look at the biomarker discovery, you don't look at all the genetic variants as your result. You want to find those ApoE, some other major, high, strong signals, right? So transfer learning is kind of way you can learn this one domain features, inform another one. And you are not perfect, but catch the 80%, 70%, or even 50% of the big strong signal. Why not? At this stage, I think, good to try. That's my answer to it. Yeah. Again, you have your answer. Everyone has a different answer. Okay.
Any other questions? No? So please join me in giving another round of applause to Dr.
Thank you.
Okay, thank you.